Hi everyone, and this is the next installment, Unit 1, DNA and the Genome, and today we're going to be looking at DNA replication. Now what we have here is what's called the central dogma, coined by Francis Crick in 1958 initially, showing you the process by which DNA ultimately codes for proteins that function within the cell. So the process by which DNA makes copies of itself is of course replication. The process of producing a messenger RNA molecule from DNA, which you've covered already in that 5, so you should already be familiar with this, is of course transcription. And the process by which that mRNA goes on to produce polypeptides and protein molecules is called translation. But for our purposes today, we're just going to have a look at DNA replication. Now, if we take this very, very short section of a DNA molecule, when it first begins to replicate, those two strands must first of all separate. And when they do separate, each of those strands acts as a template to produce another brand new DNA strand. So in this way, for each of the new double strands of DNA produced, half of that DNA comes from the original strands in the original cell. So in that way, we say that DNA replication is semi-conservative. Now, if we have a look at the requirements for DNA replication, we need a DNA strand to act as a template. We need primers, which are short lengths of RNA nucleotides, which are later converted to DNA. And they act as a start point for the DNA polymerase to attach and begin replication. We need enzymes, which in this case are DNA polymerase, and another enzyme called ligase. And we also need a nice supply of ATP, so the enzymes have got energy to build up those new DNA molecules. So if we begin with a brief overview of how DNA replication starts, first of all, the double helix itself unwinds, and those hydrogen bonds that are holding those bases together, they become weakened, and the DNA strand separates, like so. So where that DNA strand has separated, the point of separation is called the replication fork, and where replication actually begins, this is called the origin of replication. And from that point, the DNA polymerase would come along and replicate the DNA strands like so. Okay, and that whole opened section of DNA is called a replication bubble. Okay, so if we begin to look at DNA replication itself, the actual process in a bit more detail, the first thing you need to be aware of, and you need to remember this, is that DNA polymerase can only add nucleotides to the 3' end of a growing strand. So, when you see this replication fork initially opening up, a primer attaches firstly to the 3' end of the template strand, so the primer is running in the opposite direction from a 5' to 3' direction. As the replication fork opens up, DNA polymerase will come along and will begin replicating a DNA strand from that 3' end of the primer, like so. Like that, in that direction. And the DNA polymerase will do that continuously, uninterrupted. Now, for the opposite template strand, the opposite DNA template strand, only when the replication fork has opened up enough will a primer be able to attach, and so DNA polymerase, working from a 5' to 3' direction, will begin to make a copy of the opposite strand, like so. So as time goes by and that replication fork opens up even further, the DNA strand that's being copied from the left hand side of the picture here, the DNA polymerase will continue to work the way it has done, continuously building up that DNA molecule like so. But once again, it's only when the replication fork has opened up further still will another primer be able to attach to the template strand on the other side, and DNA polymerase then being able to replicate more DNA like so, because DNA polymerase can only add nucleotides to the 3' end of the growing strand. So the strand being produced on the left is produced continuously by DNA polymerase, but the strand being produced on the right hand side has to be done in sections. So the strand produced on the left hand side, produced by DNA polymerase all in one go, is termed the leading strand, and it's produced in a continuous fashion. However, the strand produced on the opposite side is called the lagging strand. It's produced in sections by DNA polymerase, and so its production is described as discontinuous. Towards the end of replication, these sections of lagging strand that have been produced by the DNA polymerase are finally joined together by an enzyme called ligase, which stitches the gaps between those different fragments of lagging strand by forming those strong chemical bonds in between those nucleotides. Finally, the RNA primers are then replaced with DNA, and the process of replication is complete. So to finish off, if we have a look at the importance of DNA itself, DNA is the molecule of inheritance, and that hereditary information is encoded in a sequence of bases along the DNA strand. This determines an organism's genotype, and the genotype plus environmental effects equals an organism's phenotype. Phenotype is an organism's physical features and also its chemical state as determined by the proteins produced by its genes and their effect on the organism's metabolism. Okay, thank you very much for your attention.